All right, this is the new neuron model as opposed to the old neuron model, which hopefully I will make a video of soon as well. Starting back on the back, we're actually looking back on this back sheet. We have several presynaptic neurons back there, and they have lots of branches, and you can see those branches going off in different places in the background. Some of them are working their way into the foreground, up through here, this flexible part. This flexible part, this one. So these are still part of those presynaptic neurons coming to our postsynaptic neuron, which is here. These axon terminals have the ability to attach. Remember, there's still a synaptic gap between them, but to the dendrites, which are the processes that allow information to come in. These are dendrites here too, or they can attach to the outside of the cell body, which is all of this material. Now the synaptic knob is the very end of the axon terminal that releases vesicles that might have something like acetylcholine, the neurotransmitter, in them to create a stimulus on the receptors on that dendrite or the cell body. Now, as we move into the cell body, there are several structures which we should recognize. We have the nucleus here. Inside the nucleus, we have these two dark regions. These are nucleoli. So in neurons, we have a few more of those than we have in a typical cell, only having one nucleolus. The difference in the ending, US to I, is singular versus plural. The fluid in here is nucleoplasm. It's not on the list, but there is an arrow to it to the lab packet if you are trying to ID that. Now, stuff inside the cell body beyond the nucleus. Up here we have this candy-colored material. That is the rough endoplasmic reticulum, which can be called Nissl substance or chromatophilic substance. And the chromatophilic is coming from the dyes that the rough ER responds to. We have smooth ER over here, and in green we have the Golgi apparatus. The functions of these are the same as they are in a basic cell. Two other items that we have in here, we have these little hot dog shaped things. These are mitochondria. The only difference with this one is it's been cut open so you can see a cross section of it. And then we also have these yellow items, which are vesicles being created here and moving down the axon that we see here all the way to the end. Now, moving away from the cell body, we get into the area where we narrow into the actual axon. So the axon has a constant diameter from here all the way to the end, but this is still wider here. And this region is called the axon hillock as you narrow down into that axon region. The narrowing down is done by neurofibrils, which are part of the cytoskeleton. We can see these lines in here helping to create that structure, although this is pretty difficult to actually ID. Moving into the axon itself. The axon, as we talked about in lecture, can be rather long. It can be close to 40 feet if you're talking about a giant octopus. So as we follow this axon all the way down and around, we get to the neuromuscular junction here with muscle. So what we're seeing here is the idea of axon terminals, a synaptic knob having the ability to release neurotransmitters onto the motor end plate. Now if we back up and we look at this part of the axon, it's been cut open and we can see mitochondria inside. We see these two cells out here. There's a gap between these two cells. So these are Schwann cells. The gap is the node of Ranvier. It is the gap between the Schwann cells. And keep in mind that's where the action potential jumps over for saltatory conduction, making the Schwann cells or myelin sheath make this a much faster movement of that stimulus down the nerve. Now as we look, these cells, remember they're like a jelly roll. They have wrapped around and around and around. And the inner layers that create that myelin sheath are very tightly packed. So this is looking at the layers here or here. The outer layer has a little bit of extra cytoplasm and any organelles in it. This is the nucleus of the Schwann cell, this yellow item on the model here. This outer layer of the Schwann cell is called the neurolemia. Myelin sheath is all the layers, individual Schwann cells. So make sure to read questions carefully on a lab exam so that you are identifying the correct material. And that is a hopefully great overview of our neuron model. Thank you.